Hello, everyone. Welcome and thank you for joining us again today for another great seminar as we continue our exciting seminar series. I am Dr. Carlos Bosquez, CEO at the Food Allergy Science Initiative, or FASI for short, and I'm excited to welcome you all to today's discussion. Today, we get to talk about eosinophilic esophagitis, a little bit different than food allergies, but highly connected. And we thought it would be an important topic, as we know a lot of people in our audience are very interested not only in food allergies but EoE as well. And for that, we have the pleasure to be joined by one of the leaders and experts in this field, Dr. Jennifer Maloney, who's the therapeutic area lead of immunology and inflammation and global development at Regeneron. And she'll be discussing about dupilumab and what we like to call the dual battle of fighting food allergies and EoE. So very excited to talk today about this. As you know, at FOSSI, our focus is to understand and intervene with food allergies. And as a leader in food allergy research, FOSSI is dedicated to advancing scientific innovation in research to transform the lives of people living with food allergies. While food allergies impact millions of people across the world, understanding of the disease and the availability of effective treatments remain a limitation, and therefore, this is why we do what we do. Before we start, I want to remind our audience of our special 10-year anniversary celebration. I really hope you can all join us. This is the event of the year when it comes to food allergies. Everything you need to know about food allergies will be discussed at this great event, and we will also be celebrating 10 years of excellence in scientific research. So we hope you can join us on October 6th and 7th of 2026 at the Broad Institute in Cambridge, Massachusetts. You can actually scan the QR code to register. Don't wait till the last minute because in the past we've had to unfortunately close registration early. So we hope you can all join us. It will be a fantastic discussion hearing from experts from the industry, academia, and the government. Um, this year, as you know, we have prepared an exciting seminar series. We've put together an exciting lineup of speakers. And throughout the seminar series, we have been discussing with experts in the field about the emerging therapies and clinical studies, looking at the success and the challenges in clinical trials in food allergies. And whether you are a patient, a clinician, a researcher, or just curious about food allergies, we hope this seminar series provide helpful information to you and your family. Before we begin, I'd like to clarify that FOSSI is a science-driven, evidence-based organization, and therefore, throughout the series, our intent is not to promote any products or companies, but instead to inform and educate our audience on recent clinical development efforts for food allergies and associated disorders, and also for you to have the opportunity to ask questions to better understand the benefits and the challenges of the different emerging options for treatments. So with that, I would like to take a moment to welcome our uh, great speaker today, Dr. Jennifer Maloney, who's the therapeutic area lead of immunology and inflammation in global development and Regeneron. Dr. Maloney oversees the direction of clinical trials in therapeutic areas of allergy, respiratory diseases, dermatology, gastroenterology, and infectious diseases at Regeneron. She presents scientific data, review and analyzes study data, and writes protocols for drug development, which is so important to be able to develop new therapies for these disorders. Dr. Maloney received her MD from Stony Brook University and completed her fellowship in allergy and immunology at Mount Sinai School of Medicine in New York City, I believe with Dr. Hugh Sampson, who most of us know from all the work in the field. Um, and so just to remind everyone in our audience that we will be taking questions from the Q&A box, and we will use those questions to prompt our discussion afterwards. So if you have any questions, please feel free to add them to the Q&A and Dr. Maloney will address them. And if we don't get to answer them, we'll try to also follow up via email. So Jennifer, it's a real honor to have you here. very excited to have this conversation. Typically, we are talking about food allergies, so it is great to talk about something that it is connected, but at the same time, is something a little different than what we've been discussing here. So, welcome.
Thank you. Thank you so much for having me. I really appreciate it. I'm very passionate about food allergies as well as EoE, so it's wonderful to be able to speak about these things.
Thank you, Jennifer. Typically, actually, just with that in mind, I always like for our audience to get to know you a little bit better, not only from your title of what you do at Regeneron, but your background. How did you get into food allergies and EoE, and what motivated you to do the work that you do?
Yeah, no, thank you for the question. So, as you mentioned, I did my fellowship at Mount Sinai, and I think as many people know, Mount Sinai is a food allergy referral center. When I was completing my fellowship, we were seeing quite a lot of EoE patients, but it was maybe it wasn't a super new phenomena, but it was becoming more readily diagnosed, and a lot of these patients were coming to Mount Sinai at the time. We quickly realized that there was a real unmet need for this population because the visits would take an incredibly long time. We didn't have a lot of treatment options. Typically, there was a need to refer patients to see the pediatric gastroenterologist. There was typically a need to have a nutrition consultation because often we were removing many foods from the diet. So we wanted to make sure that nutritionally the children were having their needs met. And as a result of seeing that this need existed, we actually put together one of the first, I believe, in the country, centers where we had sort of a multi-prong approach for these patients. And we had dedicated clinic time where patients with EoE would come, and only patients really with EoE would come, where they would have the opportunity to see the allergist, which was myself, the pediatric gastroenterologist, and the nutritionist, kind of like a one-stop shop, because we felt that really you need to have all these different appointments, but kind of bouncing from doctor to doctor, or from doctor to nutritionist was very challenging for the population, very challenging for the patients. And this way, we could at least make the process a bit more streamlined. Um, and I think that really the patients had such an impact on me because typically at the time, what we would have to do is we'd have to do food elimination diets. There weren't any therapies, truly. Even topical steroids hadn't been heavily adopted at that point. And so often we were basically recommending fairly rigid diets, sometimes elemental diets if the disease was really uncontrolled. But then we would have the opportunity to follow these patients and over time begin to add foods back to the diet and just really remember certain patients and how important it was when we were able to add some foods back that kind of created a sense of normalcy for the patients. We had one patient that really sticks out who was 10 years old. He had such severe disease, he was malnourished. We put him on an elemental diet, and for the first time in such a long time, he actually felt better. But now you have a 10-year-old child on an elemental diet, basically drinking a formula. So when we were able to add one of the first food groups we added back to the diet was corn. And for him to be able to have corn chips, which sounds like such a simple thing, but it created a sense of normalcy when he was at school that he could have a snack that was similar to what his friends were eating. And I just remember the gratitude and being able to do something for these patients was highly motivating. And then fast forward, the opportunity to be able to work on dupilumab, run that clinical program, and ultimately have a therapy that could help treat these patients was probably one of the most rewarding parts of my career to date. So, sort of a long answer to your question, but that's what motivated me.
How exciting, right? You have the opportunity as a clinician to be able to help people directly, but now also as a drug developer, as a therapeutic developer, you're actually also helping not only the people that you get to see in person, but people around the world. So really kudos to you to be able to translate a need and something that impacted you to now being able to actually generate solutions for them that would impact their lives and the lives of their families.
Thank you. Thank you. Yeah, it definitely was a full circle. When we got that first approval for dupilumab for EoE, it was very much a full circle moment.
So Jennifer, I know for our audience, we've been talking throughout this seminar series a lot of the latest clinical data in food allergies and the evolution of new therapeutics and clinical data for treatments. Today, I know we're switching a little bit to eosinophilic esophagitis. I know many people in our audience know about EoE and the connection that it's connected to food allergies. But would it be possible for you to share with our audience a little bit more information about EoE and its connection to food allergies?
Sure, happy to do so. EoE is definitely an allergic condition. It's something, all these types of conditions like atopic dermatitis, asthma, food allergy, and EoE, we consider that these patients have increased type 2 inflammation. And so it's kind of like a fancy way of saying they have these different allergic conditions. Food allergies, IgE mediated food allergies, the traditional anaphylactic type food allergies, happen in a higher proportion in patients who are atopic, and certainly patients with EoE do have a higher prevalence or co-prevalence of IgE mediated food allergies as well. In fact, we looked at this in our own trials where we enrolled EoE patients, and in our adult and adolescent trial, which was 12 years to adults, about 40% of patients also had a concomitant IgE mediated food allergy. And this number was even higher in our pediatric trial, which was a population that was 1 year to 11 years of age. So clearly, even within our own data, we saw that co-prevalence. And of course, it's been, highly reported in the literature as well. But we know that there's a link here, but they're different because IgE-mediated food allergies result in anaphylaxis when you consume the food. EoE, it's immunologically different because you don't have that immediate reaction, but instead you kind of develop this chronic inflammation if you continue to have that exposure.
That is interesting. Yes, I know we lump them together in the atopic march and the connections. But to your point, it's also as we learn more about disease, we start learning more about the differences and how populations are reacting differently. In this particular case, not the immediate response of an allergic IgE mediated anaphylaxis, but you get this long-term response that affects so many people. Um, when we think about the treatment paradigm and the options that are there, we know restrictive diets, proton pump inhibitors, steroids, and now dupilumab. And so, can you tell us a little bit more about what are the available options? What are the advantages, disadvantages for people in our audience who are parents or who are patients when they're thinking about the options and the right timing to get what options? Can you guide us a little bit more on what's available, how should they think about it, and any recommendations you may have?
Sure, sure. Happy to do so. And and just one point that I'll make, kind of related more to the last question, is what makes it very challenging for eosinophilic esophagitis is there's no great diagnostic test, right, to determine what foods are causing the disease. And this is different than IgE-mediated food allergies, where we rely heavily on our skin tests and our serum-specific IgE tests. Again, those tests aren't perfect, even for IgE-mediated food allergies, but if you have that clinical history of a reaction and you have a positive test, you feel very confident with the diagnosis. Sometimes people will have positive tests and there's no clinical history, and you can certainly have spurious false positive results. But overall, if you have that clinical history or even if you have very large skin tests and very high serum specific IgE, you feel fairly confident that the patient will react, have an allergic reaction if exposed to that food. EoE is different. We don't have great ways to diagnose the foods that are particularly causing the disease. And that's what makes it challenging, also from a treatment standpoint. So, you mentioned a few different options, and let me talk quickly about them. So, food elimination diets. Clearly, we know that if you take someone with EoE and you put them on an elemental formula like a Neocate or an Elecare, an amino acid based formula, for the most part, that will be highly effective. Um, However, that has certainly major limitations because one would have to drink quite a lot of that to get the nutrition that they need. And those formulas don't tend to taste very good. And, one of the great joys in life is to be able to eat. And so you're essentially removing that. So it is highly effective. And if someone has very severe disease, especially back when I was in practice, that's often what we would do. But then you have other types of elimination diets. I don't think that in practice anymore, people are giving such stringent elimination diets. But there are other elimination diets where you can pull certain highly allergenic foods out of the diet, milk, eggs, soy, the nuts, fish, shellfish, kind of the major food allergens that cause IgE-mediated food allergies are also at play with EoE. So if you pull the major food allergens out of the diet, and there are different variations of this, there's like a 6-food elimination diet, a 4-food elimination diet. So, and then another option would be just to remove milk because that tends to be the allergen that is most commonly associated, but often there's more than one. It's difficult, it's complicated, but there are different variations of this, and they can be somewhat effective. Some literature that I saw was, I think, in the range of about 50% effective. It's a little bit of trial and error, and unfortunately, you can kind of go by clinical symptoms to a degree, but you really need to do endoscopy to know for sure if you're removing that inflammation. And so that's quite invasive to have to do repeatedly when you're trying to do manipulation of the diet. Often, what we used to do in practice, if we were going to do a 6-food elimination diet or a 4-food, we'd pull the foods out of the diet, wait about 6 to 8 weeks, and then we'd do another endoscopy and see if there was improvement, and if there was, potentially add a food back. But that it is challenging to do that, and it does require an invasive procedure and can be quite a lot for people to go through. So then we kind of move into the different medicines that are available. Proton pump inhibitors, particularly high dose proton pump inhibitors, can be effective in about 50% of people with EoE. We don't entirely understand exactly why that is, but it's certainly worth a trial of a proton pump inhibitor to see if it's as simple as taking that medication, because that's fairly simple treatment option. So that's usually the first line, and that's actually in our trials with dupilumab. We ensured that patients were um, were not responders to proton pump inhibitors before they came into the trial. The next step could be potentially be, and it's not really steps, but the next thing that we think about are topical corticosteroids. They can be quite effective as well, particularly if a patient is very compliant with use. The downsides to that is you're essentially swallowing um, topical steroids, and so there are some side effects like candida that can happen as a result of having this exposure to the topical steroids. Um, there's one approved medication that I'm aware of right now in the United States, which is a budesonide suspension. That indication is for 12 weeks. So again, it's kind of like a Band-Aid in that when you stop it after 12 weeks, I don't believe there's any evidence that it disease modifies. There have been studies looking at prolonged use, though, and from a safety standpoint, they do seem to be fairly well tolerated. And from an efficacy standpoint, particularly if patients are compliant, they do tend to be effective. And then the last step is to consider a biologic. Right now, dupilumab is the only biologic approved, but there are other biologics that have been evaluated in EoE. The IL-5 inhibitors have been. They did not show an improvement in symptoms. They did show an improvement or a reduction in eosinophils on biopsy. Don't entirely know why they didn't show an improvement in symptoms. That remains to be discovered, I guess. There are other drugs that are currently in development. An anti-TSLP, for instance, is in development. That data should be available probably within the next year or so. So there are more treatments that are on the horizon. I have some data on dupilumab, and I'm happy to go through it if that would be helpful for the audience.
Yes, please, that would be great. That would be great to see more of that data.
Fantastic. I will share a few slides. Oops. Okay. Is there anything blocking the slides?
Uh,
Oops.
no, let's see. I think we can see them well.
Okay.
You can swap display so that if you
Perfect.
want to show them.
Is the full slide showing?
It is a full slide, but also you have the next slide showing in the corner, so that's
Oh,
not a problem.
okay.
So you can do it like that, or you can swap displays to see the entire slide. Yeah, there you go. Perfect.
Oh, okay, this is good. Okay, perfect. Sorry, it's confusing with the double screens. Okay, so just so that the audience knows my disclosures, I am an employee and a shareholder at Regeneron, so I just want to disclose that information. Um, but we evaluated dupilumab in kind of a neat study. It was one study, but we actually had a part A and a part B, which were completely unique patients. And then all patients, whether they were on part A or part B, continued in an open, not exactly an open label, but everybody got drug because there were a couple of different doses that we were evaluating. So that remained blinded, but everybody had for the last 24 to 52 weeks had the opportunity to be on dupilumab. And so essentially for part A, we evaluated dupilumab as a weekly dose versus placebo. I think if people are aware, dupilumab is approved for 8 other indications in addition to EoE, and typically the dose is 300 milligrams every other week. When we had done the initial phase 2 study for dupilumab and EoE, a weekly dose was evaluated, and that study was started at the time when we were evaluating dupilumab for atopic dermatitis, and in those initial studies as well, we evaluated both a weekly and an every other week dose. So at any rate, for the initial phase 2 trial, a weekly dose was evaluated and showed efficacy. So for the phase 3 trial, part A, we evaluated the every week dose. But then in part B, we wanted to see if the every other week dose, which was the standard dose for most of our other indications, would also be effective. And then, as I said, everybody continued on after the first 24 weeks and got dupilumab either weekly or every other week in the part for the patients that had started in part B. So these were all unique patients. And essentially, for EoE, the requirement through the health authorities is to evaluate both reduction in eosinophils in the esophagus, but also you want to show that symptoms are also reduced. And so there's a questionnaire called the Dysphagia Symptom Questionnaire that is kind of the gold standard utilized in these types of trials. And that was evaluated along with the reduction of eosinophil counts on esophageal biopsy as co-primary endpoints. And then we looked at other things additionally as secondary endpoints. So this slide basically shows the results from part A and the results from part B, but the initial 24 weeks when you had a placebo arm. We see the percentage of responders on the y-axis, and then it's just the different arms on the x-axis. So for part A, I mentioned we had a 300 milligram dose every week versus placebo, and you can see that it was close to 60% of the patients met the reduction of the eosinophil count in the esophagus. And it was actually a very stringent endpoint because they had to have less than 6 eosinophils per high power field in different parts of the esophagus. Um, and if you think about it, for the diagnosis of eosinophilic esophagitis, the diagnosis is made if you have over 15 eosinophils per high power field on that biopsy. So actually, we required it to be even lower than the actual level that you use for the diagnosis to be made. You can see similarly in part B, we showed similar effects with about 60% responders for this biopsy endpoint for both the weekly dose as well as the every other week dose. So everything looked as it should be for with respect to the improvement on biopsy. But fast forward for that other co-primary endpoint, basically the symptom endpoint, looking at difficulty swallowing, very interesting results. And so we have the absolute change in the dysphagia score on the left-hand side for both Part A and Part B. And then we have the percent change for the dysphagia questionnaire on the right-hand side. And essentially, it's just different ways of looking at the same data. But I'll call your attention to the left-hand side. And basically, what you can see here is that the every week dose had a very robust improvement or reduction in the dysphagia symptom questionnaire, but that every other week dose, which is the standard dose for dupilumab for all the other indication, did not differentiate from placebo. So we were actually quite surprised by that. We really looked into the data to make sure that there wasn't some sort of error because we couldn't really explain why this dose would have to be higher for eosinophilic esophagitis. And we still don't entirely understand why, aside from maybe you do need to have higher drug concentrations to get to the GI tract. It's a little bit unclear. But at any rate, we were very happy that we evaluated the every week dose in the study, and we were able to replicate the effect on improvement in the difficulty of swallowing. So patients had less difficulty swallowing. We were able to replicate that result across these two different patient cohorts in Part A and Part B. And we continued everybody, as I mentioned, on dupilumab. And essentially, what you can see is that the patients who had initially been put on dupilumab for the first 24 weeks had a maintenance of effect in the second part of the trial when they knew they were on active drug. But actually, you also see a robust improvement in both the symptoms as well as in the biopsy results for those patients who had initially been put on placebo and then got changed to dupilumab during that second part of the study. So again, you have some more internal replication of the efficacy within the same trial. From a safety standpoint, there really wasn't anything that was out of what would be expected for dupilumab. Clearly, when patients are getting injections, they do get injection site reactions just from having to get a subcutaneous injections. And then there were, things like some upper respiratory infections and things like that. And this shows the results for the Part A patients as well as those patients who continued on the open label every week dose for Part C. And similar data was seen for the Part B patients who were on the different doses of dupilumab. Here we just are showing the every week dose as well as when they continued on therapy in Part C. So nothing really unexpected here. Just really quickly, because I don't want to take too much time going through all of this, but we also did a study evaluating dupilumab's effect in children. And so in this study, we evaluated children who were 1 year of age up to 11 years of age. So basically to have the whole constellation of age groups. And here we also had a Part A and a Part B. It was a little bit different though, because these are essentially the same patients. So patients came in, they were randomized either to Dupixent or placebo. And at the time, we started this study when the adult and adolescent trial was still ongoing. So we didn't have that data when this study was initiated. And as a result, we had two different dosing regimens in our pediatric trial, a dosing regimen looking at lower exposure. So essentially a regimen that approximated that every other week dose for the adult and adolescents. And then we had a higher exposure, which approximated the dose that was given to adult and adolescents for the weekly dupilumab dose. And we compared that to placebo. And then similar to our adult and adolescent study, all patients continued into an extended active treatment period where everybody had the opportunity to receive dupilumab. And then there was a Part C, which was an open label extension once we had the final dose. And essentially what we saw is that, and this was also somewhat based on extrapolation because you're not able to do the dysphagia symptom questionnaire in young children. And so it was a bit of an extrapolation from a clinical standpoint that was accepted from a regulatory perspective. However, from the eosinophil count, we were able to show that we reduced the eosinophils in those esophageal biopsies for both exposures We were able, and we, because of what we knew with the adult and adolescent trial with respect to the dysphagia questionnaire, we pursued the higher exposure regimen. Similarly, uh, at the time, uh, the safety profile really looked similar to what we saw in the adult and adolescents, along with what has been seen in our large dupilumab safety database in general. So I think that's really it for the slides, but I'm happy to go through any additional data or answer any questions.
That is excellent, Jennifer, and great to see this information. We are big fans of seeing the strong clinical data to draw conclusions. So very, very helpful. Um, there's a few more questions, but I also want to be cognizant. There's a lot of I'm getting some Q&As from the chats, which is great. So maybe before we jump into more questions in the Q&A, um, I will maybe why don't we stop there? I'll just remind our audience that we always get the question of how can you help donate, host an event, follow us on social media, join our next webinar, great webinar series if you want to learn about food allergies and EoE. So please join our webinar. You can scan the QR code. And our next webinar, just as a reminder, would be by Dr. David Gold, and he'll be talking about aquestive therapeutics, which is actually now getting a very interesting option for a film for anaphylaxis. So this will be on May 5th at 12 p.m. So don't miss that webinar. Um, why don't we go to Q&A? I'll actually start with a few things that are in my mind. Uh, Food allergies, EoE, dupilumab in food allergies. It's obviously a question that I'm sure it's in everyone's mind. Proof for EoE has been attempted, there's been attempts for use of dupilumab in food allergies, and as a monotherapy hasn't been approved for that. So want to ask the expert, what are your thoughts of where dupilumab is with respect to food allergies and what may be opportunities for dupilumab for food allergies?
Sure. No, I think it's definitely on a lot of people's minds. We get asked this question all the time. And as a result of being asked the question, we did do a study looking at dupilumab. It was an open label study. It's a very small study, but enrolling patients who had confirmed food IgE mediated food allergies, confirmed by double blind placebo controlled food challenge. And they were treated for 24 weeks and then had another food challenge at the end of that 24-week treatment period. We did not show a significant effect at all, honestly, there. Only two patients were able to tolerate 444 milligrams of peanut protein at the end of that. It was because they were peanut allergic patients at the end of that study. Now, why is that? I think there are a few reasons. We do know that dupilumab does reduce IgE, but it doesn't reduce IgE to the same degree as something like a Xolair. And it takes quite a bit of time. We have, we have studies where we've monitored patients for long periods of time who've been on dupilumab who came in with elevated IgE. It takes about after about a year, your total IgE will drop about 70%. I mean, that's, around the ballpark of what we've seen within our trials. And longer durations, it will drop even more. But you're not getting to that really low level that one would need for someone who has an IgE-mediated food allergy. So potentially it could help some people, potentially, but not really. It's not going to be something like a Xolair that's going to make it so that you would feel more confident that you wouldn't react to an accidental ingestion. So where does that leave us, and why is that? I mean, we have some theories as to why that is. We do know that if you use dupilumab and you're blocking IL-4 and IL-13 because you're blocking the IL-4 receptor alpha with dupilumab, so you're blocking those cytokines, we are putting an end to class switching. So you're basically preventing the generation of new IgE when you're on dupilumab because you're blocking that pathway. But there are already plasma cells that are long-lived plasma cells that are making IgE, and we don't do anything to remove that source of IgE. So particularly for people who have longstanding food allergies, they already have those long-lived plasma cells. Something that we're currently evaluating is what if you eliminate those long-lived plasma cells? Can you then potentially, with a background of dupilumab, uh, cure food allergy or provide a potential cure or do something to prevent someone from being able to react? And we have a study actually ongoing looking at this. It's a very small proof of mechanism study, but essentially, patients with very severe IgE-mediated food allergies are participating. They come in after and they receive a background of dupilumab, so they're on dupilumab for at least 12 weeks, and then they're given a short course of a medication called linvoseltamab, which is actually a medication for multiple myeloma, but it obliterates or ablates the plasma cells. And so with that, in theory, you should be able to remove that source of long-lived plasma cells that are producing IgE. Then you remove that medication. It's only a short course, and your plasma cells will repopulate, so you don't lose your long-lived immunity. However, with the background of dupilumab, you shouldn't be able to class switch, and essentially, you should be able to have, a treatment option that could potentially eliminate the IgE-mediated food allergy. We're still in very early days, but we do have some animal data that supports this. And we do have a study ongoing in very severe food allergic patients.
That is interesting. Thanks for sharing that information. It's a question we often get about why dupilumab didn't actually show response as a monotherapy, and the difference with Xolair and the decrease of IgEs was not sufficient. Is that the only answer? But great to hear that Regeneron's looking at other additional trials to better understand that system. Um, Question about there are about 30% non-responders to the dupilumab, if we read it correctly from the data. Any thoughts of why those are non-responders, and what are the options for that population that do not respond to the dupilumab?
Yeah, and I mean, I'm not sure if I'd say they're entirely non-responders, but maybe they're partial responders because you do notice that as you continue on treatment, your reduction in esophageal eosinophils will continue to reduce. Like so by week 52, you can be close to 70, 80% having that reduction. Again, we don't have a placebo arm there, so it's hard to compare it to placebo, although looking at just esophageal eosinophils, it's fairly objective. Um, but clearly, there are some patients that don't respond and or don't respond adequately. I think, this is why it's great to be in a field where there is continuing research. As I mentioned, there are other medications currently in development. It would be very interesting to see how people respond to that medication. Does it treat a population of patients that dupilumab doesn't? I mean, we don't know. The data is not available yet. It's not going to be a head-to-head trial either, but it will be interesting to compare the efficacy when available with other agents out there. Um, I think there are still unmet needs for sure. Clearly, dupilumab is at this point, we believe it's something that one needs to stay on. We don't have evidence that it's disease modifying. We've never evaluated it. It's something that would be great to evaluate in the future if there is any disease modification that happens. From the data that we have right now, there's nothing to support that it would be, although it hasn't been evaluated. So there's nothing to support, I suppose it wouldn't be. But I wouldn't expect, based on mechanism, that it would be disease modifying. I think also, just in general, it would be really great to understand what is causing eosinophilic esophagitis. I mean, we have some ideas, but again, it's like a lot of diseases that are increasing in prevalence. We have some ideas. For instance, the inflammatory bowel disease seems to be increasing in prevalence. And why is that? There's a connection between epithelial barrier and these conditions with EoE. There's probably something related there. I mean, these diseases are not considered to be closely related, but there are elements that are quite similar. Um, you know, again, it's not backed on data on research that I've done, but I think there's something environmentally that must be happening because we're seeing this increase in prevalence of a lot of these different diseases that were just not so common 30 years ago. So understanding why this is happening and what can we do to prevent these different diseases from occurring would be really important for the future of research.
Excellent. Yeah. No, there's a lot of unknowns that would be important to understand. I always feel that, clinical trials are important not only for developing the therapy, but also to get additional data that leads to further understanding in the most important population, and that leads to better therapies in the future. So this is very helpful. There's one question here that I'll actually mix with something you said before, which comes to diagnostics, limited diagnostics in EoE, and the need of endoscopies to be able to diagnose. Um, why is so difficult to diagnose? What are the limitations? What are the options that you have seen already in development that could overcome that challenge? One of the question here is AI. Is there any role for AI to help replace the gold standards for for diagnosis here? So thoughts on diagnosis of what's missing, what's there, and what's missing.
Yeah, I mean, unfortunately, I think for a lot of these conditions, we have many limitations to diagnosing, right? I mean, for IgE mediated food allergies, the gold standard is the oral food challenge, the double-blind placebo-controlled oral food challenge, which is also um, really time-consuming, stressful for patients, stressful for everybody. I mean, stressful even for physicians if you have someone that has had anaphylaxis before. I mean, it's, you want to be able to introduce foods to the diet and you get to a point where you feel like maybe you can, but there's always that risk. And so it's it's definitely a lot to have to put someone through. And as you mentioned, EoE, the endoscopy is really at this point still incredibly important to get those biopsies and to see what the eosinophil counts are. And even when you're treating the patient to ensure that you're having a remission of disease, because leaving the disease unchecked clearly has long-term ramifications with respect to having fibrosis of the esophagus. And so you clearly want to make sure whatever intervention you're using is actually working. I mean, I'm not an expert in AI. I mean, it seems like a very powerful tool. That would be amazing if there's something in the future that could help us with this. And I know clearly that a lot of researchers are working for new types of biomarkers and new types of ways to try to make these diagnoses in a more straightforward way for both patients and physicians. I mean, at this point, I think we have the tools that we have. Definitely, there are advances being made, but we're just not there yet. Oh, I think you might be on mute.
Yes, I was on mute. Thank you. No, thank you, Jennifer. It seems like there's clear unmet needs in EoE, not only in great work to have some therapeutic options, but also needs in terms of diagnosis. To your point, if you could actually try to get it earlier, detect it earlier to prevent any fibrosis, which is a more difficult challenge afterwards, those would be great. So. Um, for everyone that is listening, that's an area hopefully that people can do more work and help Regeneron do what they're doing with the great therapy. So um, we're only 1 minute left, so I would just I want to close it here. I think there's a lot of great questions we have here. Hopefully, we can address them separately. But want to take the time to thank you, Dr. Maloney, for joining us, for spending an hour of your time educating us about EoE and the options that we have now with dupilumab and all the other opportunities for people to use in the treatment paradigm. It's really an honor to have you and thank you.
Thank you so much. I so enjoyed this presentation and this discussion. Thank you so much.
Thank you. Thanks everyone for joining today.